Good morning, all of you. In last practical, I have discussed to carry out the isolation of culture by use of pour plate technique. Today, we will see the next practical, that is to perform the sterility test for sodium chloride injection. We will see what this practical, that is to perform sterility test for sodium chloride injection. First, we will see introduction about this practical. The sterility test is carried out to detect why it is carried out or why it is essential, because to detect the presence of viable forms of microorganism in pharmaceutical preparations, those which are required to strictly sterile. Then the large number of pharmaceutical preparations are required to be sterile, such as ophthalmic, then injectables, then implants, surgical instrument, dressing, then syringes and needles. Then such preparation, it must be sterile or it must be free from any kind of microbial contamination. Therefore, it is called as sterile products. As you know, the meaning of, as I discussed earlier, what is sterile, that is, it means it should be free from all type of microorganism, that is sterile. The sterility test is one of the evaluation tests for sterile products. Yes, sterility test is one of the evaluation tests for sterile products. Then it is based on the principle that if microorganisms are kept in nutritive media at favorable temperature, microorganism will grow, and their presence can be detected by turbidity in original clear medium. Then we'll see culture media. In culture media, first fluid thioglycolate media, then second alternative thioglycolate media, and third soybean casein digest media. First, fluid thioglycolate media, it is used with clear fluid products. Then alternative thioglycolate media, it is used for turbid or viscous products. Then it is used for turbid or viscous product and devices having tube with small lumen. Then soybean casein digest media, it is used to detect the presence of fungi. Then the pH of media is adjusted 7.1 plus minus 0.2 and it is sterilized. It means this culture media are sterilized by the use of moist heat sterilization. Yes, in moist heat sterilization, it means there is use of autoclaving and at 121 degrees Celsius for 15 minutes at 15 IBS pressure. Then next one, that is sterility of media. The fluid thioglycolate media and alternative thioglycolate media, these both media are incubated are at 30 to 35 degrees Celsius, and third media, that is soybean casein digest media, it is incubated at 20 to 25 degrees Celsius for a period of 7 days and observe the presence of growth of microbes in media. Then growth promotion test, it is carried out to check nutritional quality of media. This growth promotion test, it is carried out to check nutritional quality of media. Then 100 number of viable forms of microorganism of strains as listed in table are inoculated to media and observed after incubation period of 7 days, and the media is satisfactory. If clear evidence of growth is appear, then this media can give a reliable result. We will see this table shows the medium, test microorganism and incubation temperature and the incubation condition. First media, that is fluid thioglycolate test organism, that is Bacillus subtilis, then Candida albicans and Bacteroides vulgatus. For Bacillus subtilis, temperature required for this 30 to 35 degrees Celsius and condition it is aerobic. Then for Candida albicans, temperature it is 30 to 35 degrees Celsius and condition it requires that is aerobic. Then Bacteroides vulgatus, then temperature required for this 30 to 35 and condition that is it requires aerobic condition. Then next, alternative thioglycolate media. The test microorganism that is Bacteroides vulgatus and temperature 30 to 35 degrees Celsius and condition it is anaerobic condition. Then soybean casein digest media, test organism that is Bacillus subtilis and Candida albicans. Incubation temperature required for this 20 to 25 and condition it is aerobic. Then for Candida albicans, temperature required for this it is 20 to 25 degrees Celsius and condition required for this it is aerobic. Then test for bacteriostasis and fungistasis. Then this test is carried out to detect the presence of any kind of fungistatic or the bacteriostatic agent if it is present in sterile a product which is subjected for sterility. The culture of bacteria and fungi is prepared as different strain which are seen in above table. Then inoculate the media with 100 number of microorganisms and add specific amount of sample of sterile products to the media. Then incubate the media for a period of 7 days and observe the growth. Then if clear growth is observed, then it indicates that the sample under test it does not contain any fungistatic or any bacteriostatic agent. Such sample can be directly used for sterility test. Then if clear growth is not observed, then it shows that if the clear growth is not observed, then it shows that sample contains fungistatic or the bacteriostatic agent. Therefore, suitable neutralizing agent to the sample should be added before conducting the sterility test. Then we will see the dilution fluids. There are two types of dilution fluids are used. First, fluid A and second that is fluid B. In fluid A, dissolve 1 gram of peptic digest of animal tissue that is bacteriological peptone in water and make a 1 liter. Then filter the solution, then adjust the pH that is 7.1 plus minus 0.2 and sterilize this fluid of the media at 121 degrees Celsius for 20 minutes. Then second that is fluid B. If the test sample contain lecithin or the oil, use fluid A and add 1 ml of polysorbate 80 to each liter of fluid A. Then filter and adjust the pH that is 7.1 plus minus 0.2 and sterilized it at 121 degrees Celsius for 20 minutes. Then this is all about dilution fluids. Next, that is method for sterility test. Method A, that is membrane filtration method, and method B, that is direct inoculation. We will see one by one. First, that is membrane filter method. Use membrane filters having a nominal pore size not greater than 0.45 micrometer, in which the effectiveness to retain the microorganism has been established. Then cellulose nitrate filter, for example, are used for aqueous, oily and weakly alcoholic solutions. And cellulose acetate filters, for example, are used for strongly alcoholic solution, and specially adapted filters may be needed for certain products, for example for antibiotics. Then second method, that is direct inoculation. In this method, the transfer the quantity of preparation to be examined in suitable amount directly into the culture medium, so that the volume of the product is not more than 10% of the volume of the medium, then unless otherwise it is prescribed. If product to be examined has antimicrobial activity, to carry out the test after neutralizing this with a suitable neutralizing substance or by dilution in a sufficient quantity of culture medium. When it is necessary to use the large volume of product, if or it may be preferable to use a concentrated culture medium prepared in such a way that it takes into account the subsequent dilution, where the appropriate or the concentrated media may be added directly to the products in its container. Then we will see the requirements of this practical. It requires media, that is then requirements of this practical: first, media, that is fluid thioglycolate medium, and second, soybean casein digest medium. Then apparatus required for this practical: test tube, conical flask, glass marking pencil, forceps and scissor. Then equipments: the laminar airflow, membrane filter unit, vacuum pump, incubator and autoclave. Then we will see the principle of this practical. The sodium chloride injection does not possess any this sodium chloride injection, it does not possesses any antimicrobial activity. Therefore, the inactivation of given preparation is not required. And total volume of each container is 500 ml. As per guidelines are provided. Method A should be used for assay of sodium chloride injection. It is given that whenever the volume in container is 100 ml or more, total volume should be filtered through membrane filter. Then we will see the procedure. First, clean the membrane filtration unit. Then second, sterilize the unit and membrane filter separately by the use of moist heat sterilization, that is by using autoclave. Then third, transfer the unit on laminar airflow bench or aseptic area and place membrane filter in the unit. Then filter all solution through the membrane filtration unit under vacuum. Then after filtration, the membrane is removed aseptically and cut into two parts by use of sterile scissor. Then 1/2 part of membrane filter is placed in 100 ml of fluid thioglycolate medium and incubate at 30 to 35 degrees Celsius for not less than 7 days. Then other half part of membrane filter is placed in soybean casein digest medium and incubate at 20 to 25 degrees Celsius for not less than 7 days. Then what are the observations and result? Then observe and compare the turbidity in medium with standard tube. If there is no turbidity in fluid medium and soybean casein digest medium, it is clear that the sample under test does not contain. It means it does not contain any type of or any form of microorganism and it is suitable for use. Then the such preparation can be called as sterile preparation. If any type of turbidity is found, then repeat the experiment for confirmation of result. I am repeating the observation and result. Observe and compare the turbidity in medium with standard tube. If there is no turbidity in fluid medium as well as soybean casein digest medium, it is clear that the sample under test does not contain any form of microorganism and it is suitable for use. And such preparation can be called as sterile preparation. If any type of turbidity is present or if any type of turbidity is found, then repeat the experiment for confirmation of result. So this is all about this practical, that is to carry out or to perform the sterility test of sodium chloride injection. I will stop here.